Hi, everyone, and welcome to the Critical Path Institute's Rare and Orphan Disease Program webinar series. This one's hosted today by the LGMDs Task Force. My name is Dominique Cruz, and I'm a project manager for the Rare Disease Cures Accelerator Data and Analytics Platform, or RDCA DAP for short. RDCA DAP provides a centralized and standardized infrastructure to support and speed up rare disease characterization with the goal of accelerating therapy development for rare diseases. It's my pleasure to welcome everyone to today's webinar, The Power of Data Sharing in Collaboration Across LGMDs in a Pre-Competitive Neutral Environment. Before we begin, I'd like to go over a few housekeeping items. If you have any questions throughout the presentation for our speakers or panelists, please put them in the Q&A box. The chat function is disabled for everyone, just so we make sure we don't miss any questions that come up. And all the participant lines are muted so we can reduce background noise. We are recording this presentation and it will be available on the CPAP YouTube channel for on-demand viewing. And we also have our contact information there, and we'll display it again at the end. So just for a quick run of show, today's webinar, as I mentioned, is to discuss the newly launched LGMDs Task Force, a new effort convening several stakeholders to tackle some unmet needs in the LGMDs community. We will start with a presentation from Dr. Straub, followed by a quick overview of the task force by Dr. Grabenstatter, and ending with a discussion and Q&A with a group of expert panelists. Now I'm pleased to introduce our first speaker, Dr. Volker Straub. He's the Harold Macmillan Professor of Medicine and Professor of Neuromuscular Genetics at the Institute of Translational and Clinical Research at Newcastle University, United Kingdom, and the director of the John Walton Muscular Dystrophy Research Center. Dr. Straub, I will hand it over to you.
Well, thank you very much, Dominic, for the kind introduction. I start with this slide because I think it's the first time that I have seen my webinar actually promoted through social media and LinkedIn. And I also took a close look at the title, The Power of Data Sharing and Collaboration Across LGMDs in a Pre-Competitive Neutral Environment. We always ask ourselves, does this environment really exist? And looking at the title, I thought I'll divide the talk into two parts and then bring the two parts hopefully together. So just to get an understanding of what data sharing really means and what's the power of data sharing. And then, of course, getting the limb-girdle muscular dystrophies in there and how these two topics fit together. So if you go to the next slide, if people think that browsing the internet, for example, is free, then they may not have figured that you're actually paying with data. And I think many of us know that data is a new currency now in the world, and it's particularly, of course, about big data. The more data you have, the more valuable they become. And data analysis is now, of course, making more and more use of smart technologies, machine learning, or artificial intelligence. So this is just the new world I think we are living in. And so data sharing becomes more and more important on a large scale as well, particularly if you deal with rare diseases where you need actually data from all over the world to have a large data set. So I just want to focus on some of the concepts of data sharing. So the next slide, please. And this is really a more general introduction. What does data sharing really mean? Data sharing refers to the sharing of data among diverse stakeholders often. I mean, you can share data internally within, for example, a university, but more often we refer to data sharing in terms of external and various stakeholders. Now, we distinguish between, and this is a bit business terminology, I think, but it shows how it has entered, the business world as well, between data producers and data consumers. And data producers, of course, need to make sure that their data are of high quality, so they carefully need to document and label sets of data. And this is important so that data consumers, can make sure that data is retained in its high quality, it's coherent, it's useful for the processing and analysis by the data consumers. And I think, everyone has already experienced this in your everyday life as well, that, robust access controls and permissions need to be implemented, if you want to access bank data, for example, but also, of course, patient data, personal data. We need to make sure that access to data is robustly controlled. So there are now principles that we apply to make sure that these concepts of data sharing actually work. And the next slide, please. A group of those principles are the FAIR data principles. FAIR stands for findable, A for accessible. So where do we store data? How can you access them? And if you have different data sources in different databases, are these databases interoperable? And ideally, we want to make sure that data are reusable, which has been a big challenge in the past for clinical trials, for example. Finding these data, reusing them, making sure that they're interoperable. So the FAIR data principle is something that we now try to apply more and more, for example, in our patient registries as well. So the next slide, what makes data sharing often challenging is that data are of value. I pointed out, data is the new currency. And the value of data depends, of course, on the time that you have to spend to generate them. Also, the training of people, the qualified people that you need to generate data, the infrastructure, if we think about, for example, data generation in a clinical trial, the equipment and personnel, patient, if we store data that are, individual, but also, data from carers in our field. And I've just shown this diagram with the scale there because we need to be aware that in rare diseases, of course, the higher the number of patients, the better, but often we only get high numbers from, in real world data in a clinical setting, maybe patient registries. But once it becomes interventional clinical trial, the patient numbers decrease, but data robustness, data quality is often better. So you can either have a lot of data with maybe slightly less quality or very few data with a very high quality. And the next slide also shows, harnessing the power of data sharing. So the benefits are often very clear. You want to unlock insights, which means, the more data you have, the easier it is to find trends, for example, what data in a clinical trial or a natural history study, or even a patient registry might reveal. Uncover hidden trends. Again, the more data you have, the easier this is. But it's very important there's a spirit of data sharing which actually drives collaborations as well. We want to accelerate results and ideally fuel growth. Now, I've already mentioned some of the challenges that, how do we ensure data security? How do we ensure data privacy? And this is why we are now facing more and more regulations that we need to meet that protect data security and data privacy. And this is challenging because we have to navigate regulatory compliance here. Of course, a huge challenge is massive volumes of data. And thinking about rare diseases, this is, for example, for us, particularly if we think about sequencing data, not always easy to transfer data, which, again, coming back to FAIR data principles, might also be less and less relevant if we know where to find them, how to access them, making sure they're interoperable and reusable. And then, of course, the recognition of data producers and the value of data. And I think working with academics, that is often a challenge. I mean, if we have a grant that supports data collection and someone else then wants to have the data, we say, wait a minute, I mean, these are our data. We don't give them away lightly. It's easier to give away a score for a patient assessment, for example, but when it comes to complex data like imaging data or biomarker data or sequences, this is a challenge. So coming to the next slide, this was a bit of an introduction about the concepts, benefits, challenges of data sharing, best practices. So best practices really mean that, and this is important for academics, I think, to really consider when they work also with industry. We have to implement clear governance policies. Who is responsible for the data? Who do we need to involve? Data transfer officers, for example. Define data sharing protocols and implement a data exchange platform, which in the business world is often referred to a marketplace. And I mentioned the data exchange in the platform, applying fair data principles, doesn't necessarily mean that you need to transfer data when you share data. So this was the introduction to the data sharing. I'll now come to the limb-girdle muscular dystrophies, and I'll also start with a slight definition. And I use a definition that that's a group of key opinion leaders in the limb-girdle muscular dystrophy field, including patient advocacy groups, came up with at a workshop of the European Neuromuscular Center that was specifically aimed at looking at the classification and the nomenclature of limb-girdle muscular dystrophies. And we realized that, first of all, we had to actually agree, well, what do we understand the limb-girdle muscular dystrophy actually is? And so we said limb-girdle muscular dystrophy is a genetically inherited condition that primarily affects skeletal muscle, leading to progressive, predominantly proximal muscle weakness at presentation caused by a loss of muscle fibers. And if you animate this slide, then you will see that we have defined this a bit more precisely, which says that we think it's important for a genetic condition to at least have two unrelated families. Otherwise, you are never certain whether a variant is really responsible. And then we felt it's important that individuals with limb-girdle muscular dystrophies must have achieved independent walking to really distinguish them from congenital muscular dystrophies. They must have an elevated serum creatine kinase activity, must demonstrate degenerative changes on muscle imaging over the course of the disease, and having dystrophic changes on muscle histology, ultimately leading to end-stage pathology for the most affected muscles. And I've illustrated this on the next slide, which basically summarizes what I've just said. So it's a progressive muscle wasting disease, the primary damage of muscle fibers. The shoulder and pelvic girdle are primarily affected, and you can see that on the muscle biopsy, you may start very early on, even pre-symptomatically, with a fairly normal biopsy, and ultimately this leads to degenerative changes in a replacement of muscle by fat. This is also illustrated in the muscle MRI, where you see a cross-section through the thighs. And so what's white is basically subcutaneous fat, or the bone marrow within the center of the bones, of the thigh bones. And muscle normally has a dark grayish signal on these T1 weighted images. And over time, this is a boy with Duchenne muscular dystrophy in this case, but it's the same. We see this in limb-girdle muscular dystrophy as well, but the muscle is replaced by fat. So that's the definition. Now, how many limb-girdle muscular dystrophies are there? If you go to the next slide, this is often, they're all genetically determined. This is often a slide where, you lose the audience because you think, oh God, this is overwhelming. And this is really something for the limb-girdle geeks to, go through this long list of diseases. Now, despite the fact that we have a lot more than 30 forms of limb-girdle muscular dystrophy now, it's important to understand that many of them are ultra rare. And actually, when I say ultra rare, there may only be a few families that have been described in the world. We classify the limb-girdles based on their mode of inheritance. D stands for dominant, and R stands for autosomal recessive mode of inheritance. And then the number is just in terms of the historical description. So R1 was the first autosomal recessive limb-girdle muscular dystrophy that was described based on genetics, and limb-girdle muscular dystrophy D1, the first dominant. Now, the nomenclature and the classification has changed over the years, and it is difficult to find a solution that pleases everyone. Generally, the recessive ones are a lot more frequent, about 90% of all limb-girdle muscular dystrophies, and the two most common ones are actually R1 and R2, caused by mutations in the Calpain 3 gene and in the dysferlin gene. The CK is generally high in the recessive ones and lower in the dominant ones. So now this is just an overview, and if we come to the next slide, the question is now, how does it refer to data? Well, we do now have quite a lot of patient registries for specific forms of limb-girdle muscular dystrophy. You can see the link here. And of course, the limb-girdle muscular dystrophy patient registry provide a wealth of data as well. Now, they are set up by different organizations, and they have not been set up following clear, fair data principles. So interoperability is difficult, and we therefore need to also find solutions. How can we make really use of this data to learn something, maybe some principles that apply to all of them? But it's great to see that there is a large number now of patient registries. Coming to the next slide, please. And as you may recall, when I talked about data sharing, I showed that, well, data from clinics are, of course, most abundant, and then patient registry, and then natural history study, the data robustness increases. And this is a summary slide from a workshop report that specifically focused on natural history studies in limb-girdle muscular dystrophies. Despite the fact that this is now already 3 years old, it's more to show you that there is a lot of activity. The different studies are listed on the top of the slide. So there's the GRASP LGMD consortium, which actually looks at a number of limb-girdle muscular dystrophies. There's a natural history study from colleagues at Columbus, Ohio, and also the Iowa natural history study for patients with LGMD R9 or 2I caused by FKRP. There's a 2I consortium that we were leading a few years ago. Geneton natural history study to also lead into a gene therapy trial. There's an LGMD2A consortium. Well, I'm not going through all of them, but I do want to focus briefly on the Jane COS consortium, because I will use this as the biggest natural history study. You can see here that we recruited more than 200 patients, and we followed them up for now over 10 years. And so we looked also which data sets are collected, and what became quite clear, particularly if we look into the physiotherapy assessment. Can you go to the next slide? Where you see a bit more granularity about how patients were assessed, that there weren't two natural history studies that actually collected the same data. What is good to see is that more and more of the studies are now talking to each other as well, and are agreeing on what the most important data are. But it took some time to actually find out which are the most important data. And again, if you just look at the last two columns here from the Jane COS consortium 1 and 2, you can see that we got rid of some of the assessments, and I therefore want to focus a bit on this. So how do we make sure that the data that are collected are actually meaningful and valuable? And so if we come to the next slide, this is just to outline what the clinical outcome study for dystrophinopathy really is. It was, a clinical expert group that came together to discuss which assessments were appropriate. And of course, these are primarily physiotherapists who do the assessments. And then the functional measures were chosen in the absence of any suitable disease-specific assessments at the time when we started the study. Muscle strength tests were chosen based on the pattern of weakness, of course, primarily looking at limb-girdle weakness, so the shoulder girdle and the pelvic girdle. And then the idea was to collect data for the first year of the study and then to objectively evaluate the efficacy of the assessments. And then identification of assessments to continue with and whether other assessments needed to be added in. So if we go to the next slide, this just gives you a geographic spread of the COS sites. So this is a global study with more than 50 sites from 8 countries in the world. The study was funded from the beginning by the Jane Foundation, but it's really important to point out that the Jane Foundation wasn't just or isn't just a funder, but also a very close partner involved in many of the decisions and discussions for the study. So what I had just summarized in the previous slide can now be seen in the next slide, where we listed the different assessment methods. So for COS1 and COS2, and you can see that we were looking at respiratory assessments, strength testing, muscle function testing, the time test, patient-reported outcomes, quality of life outcomes, cardiac assessments, muscle MRI, and also biobanking. And we learned from COS1 that actually we don't need to do all the different measures because within a year they were either not really, or actually over the 3 years, not repeatable consistently. And we also then saw, well, there was no specific limb-girdle assessment at the time, and the COS study helped us to come up with the North Star assessment for what we initially called dystrophinopathy, but now we say the North Star assessment actually for limb-girdle like muscular dystrophies. And I'm very pleased to say that most of the interventional trials if not all actually in this field are using the NSAD as a main outcome. So the natural history really served to look at which data are important to collect over time and which data sets might actually be less meaningful. So this is just a split up again of the patient numbers and the sites over the COS study now. As I said, first patients were recruited more than 10 years ago. So we now have longitudinal follow-up for a lot of patients for almost 10 years. The age range you can see is from the mid to late teenage years up to the oldest patient was 80. Most patients are ambulant, about a third are non-ambulant. In terms of gender, it's fairly even, as you would expect for an autosomal recessive disease in terms of females and males. And we have a broad variation of disease severity, mainly based on how long the disease has already been experienced. Now, a lot of the data, and I think this is extremely important for the academic field, but also ideally, I think for companies that are doing clinical trials, is to publish data. So in COS, and that's the next slide, and this is just 4 papers that were selected, but we have published a lot more now. So, first the 3-year data was a new motor scale. This is when we introduced the North Star assessment for dystrophinopathy or limb-girdle-like muscular dystrophies. We have 3-year quantitative MRI data that we published. We looked at strength testing modalities and also at patient reported outcomes and how they link or correlate with the functional status in terms of strength. So it's really important to publish all of this. What have we learned? This is on the next slide, the insights that we gained from this large study. So we described outcomes that are most sensitive in subgroups of dysphotopathy patients and noted that progression rates did not differ really by clinical diagnosis. So for example, also Miyoshi myopathy, which has often been labeled as a more distal form, is actually, in terms of the assessments, showing the same patterns as Limb-Girdle Muscular Dystrophy R2. We described the MRI pattern, and again, actually, there was no difference between Miyoshi myopathy patients and LGMD R2 patients. Described respiratory and cardiac involvement and provided recommendations on care here. Fortunately, cardiac involvement doesn't really seem to be a big clinical issue. We described the relationship between commonly used patient-reported outcomes and the functional status, and it's great to see that the so-called active limb really correlates very closely with the status scored by the North Star assessment for limb-girdle-like muscular dystrophies. And then we focused also on the late onset disease and provided evidence, and I already mentioned this, that there is no distinction clinically between Miyoshi and Limb-Girdle. And this is really important if we think about drug approval or even clinical trial inclusion criteria. So we wouldn't want a drug to only be approved for Limb-Girdle Muscular Dystrophy R2 and not for Miyoshi myopathy patient, or that they are excluded in terms of inclusion criteria. No, they're all the same. We provided evidence that water T2 could predict functional decline in patients as well, which is really important to have a non-invasive biomarker or surrogate marker. We also looked at biomarkers in more detail and suggested the use of myostatin and follistatin as monitoring and prognostic biomarkers might be an option. And then I think the main outcome was the NSAD. And finally, so we compared common strengths methods. Now, this has been an enormous achievement because many, many groups helped. And when it comes to data sharing, I just want to emphasize this is where the governance is important. That's where you have to have protocols in place. How do you agree that you share data? Of course, companies will know this when they have many trial sites involved. Special thanks go here, if you could just animate, to Heather Hilston, who is the senior project manager for the study. Without her, it would have been impossible. But thanks also go to the Jane Foundation, if we can go to the next slide, and of course, all the investigators and patients and physiotherapists. But the Jane Foundation also has had the foresight to actually say, yes, we are missing drugs right now, but hopefully having good natural history data might incentivize also companies to look at drug development for these patients. We go to the next slide. Just thinking about the value of data across limb-girdle muscular dystrophy, and I just want to introduce this concept of a basket trial, which originated from oncology trials for drugs that targeted a tumor mutation present in different tumor types. And so each sub-trial was testing the drug in a different tumor type. This is the history of the basket trials. So, but this can also be used for other diseases. So for the limb-girdle muscular dystrophies, I mentioned that ultimately we see muscle degeneration, no matter what the primary defect is. So if you have a drug that targets downstream effects of muscle degeneration, might this be actually applicable to, many forms of limb-girdle muscular dystrophy? So could you include many patients with different forms? And we have had this actually once before. If you go to the next slide, it was one of the very first commercial trials ever in muscular dystrophies, sponsored by Wyeth at the time, and with Kathryn Wagner being the lead for the study. And so this was a phase 1/2 trial of an antibody against myostatin. So the idea was to induce muscle growth. And I don't want to go into the study here, but I just wanted to go into the subjects that were enrolled. So there were patients with Becker muscular dystrophy, with facioscapulohumeral muscular dystrophy, or one of the following forms of limb-girdle muscular dystrophy, 2A, 2B, 2C, 2D, 2E, or 2I. And the idea was it doesn't really matter which form you have, as long as we can prove that you can induce muscle growth. Now, if the trial would have shown a clear benefit, you would know about it, but it didn't, unfortunately. I just wanted to use it as an example that, yes, having a lot of data may actually allow you to do this more effectively also for pooling limb-girdle muscular dystrophies, or for doing, for example, basket trials. So the next slide, just to repeat myself, data is a new currency. And I just, for the last two slides, want to show two projects where we are now actually also applying AI. One of them is an EU-funded project called COMPASS NMD, which means computational models for new patient stratification strategies of neuromuscular disorders, and this could therefore also apply to limb-girdle muscular dystrophies. And what our consortium does, in close collaboration with computational scientists, is to pool data from genetic sequencing, from muscle imaging by MRI, and also from muscle biopsies, so histology data. And we use algorithms and artificial intelligence to support and ideally accelerate the diagnosis of diseases. So it's very interesting to see that there are now projects that really make use of big data sets, applying artificial intelligence. And the next slide just shows a different, also EU-funded project, which is called Project Paladin, the Patient Lifestyle and Disease Data Interactium. And what the Interactium is really a space, a digital space that will make use of various data sources, including real-world data, patient-reported outcomes, registry data, to help, hopefully, decision makers like regulators, but also patients themselves, to better understand diseases, how real-world data could, for example, support registry data or natural history data. So this is just an illustration of how we are starting to use big data now also in big consortia. And with this, I want to come to the next slide and thank you all for your attention. The idea was to just get an introduction and the concept of data sharing in the limb-girdle muscular dystrophies, which is hopefully something that the task force that we will hear more about can help to facilitate as well. Thank you very much.
Thank you, Dr. Straub, for that informative presentation. So the next part of the webinar will be hosted by Heidi. So Dr. Heidi Grabenstatter is the Scientific Director for RDCA DAP, with previous academic and patient advocacy experience that greatly benefits her in her current role at CPATH. And I'll hand it over to Heidi.
Thank you, Dawn. CPATH is a nonprofit that's funded by the FDA, and the RDCA DAP platform is both a program and a data analytics platform within CPATH. The primary goal for the platform is to be a neutral, independent data collaboration and analytics hub to promote the sharing of critical data across rare diseases in order to accelerate the understanding of the disease progression and optimize clinical trial design. The way this is done is to ingest multiple types of data, as Dr. Straub had talked about, both clinical trial, registry, natural history, genomic imaging, preclinical data, and we're expanding to look at other types of data. We use responsive curation and fair data practices so that the data can be harmonized and reused by outside users and CPath quantitative sciences to develop actionable solutions. And it's governed in such a way that the contributors own their data and can select how they share with the platform and the task forces that are developed, or with any outside user that enters the CPath platform via workspace, and it could be industry or other academics that are looking to use the data, or regulators. Next slide, please. So the data that's aggregated and curated is then leveraged using the core competencies within CPath, our regulatory science team, our data standards and science team, our COA team, and biomarker team to develop tools to overcome bottlenecks in the drug development process. And the goal is to have actionable solutions for diseases like the limb girdles. Next slide, please. This webinar is, we're here to celebrate the launch of the LGMDs task force, and we've been meeting quarterly since June when we first launched as a public-private partnership convening foundations, KOLs, and industry with a vision to standardize and integrate the limb-girdle muscular dystrophy data on the platform, optimizing clinical trial design and endpoints to accelerate the development of treatment for LGMDs, and enhancing communication and collaboration amongst diverse stakeholders to improve the lives of those affected with LGMDs. Currently, the members include BridgeBio, Coalition to Cure Calpain, 3 Cure LGM2I Foundation, the Jane Foundation, LGMD Awareness Foundation, LGM2D Foundation, LGM2I Research Fund, Parent Project Muscular Dystrophy, a patient that had been working with the DMD space learned that he had actually a diagnosis of LGM2E and is sharing his patient perspective with us. The Dion Foundation, the Speak Foundation, University of Iowa, and then of course Critical Institute. And we are working across industry, those patients, the regulators, NIH, and academia as we continue to onboard new data and new members. Next slide, please. So the deliverable, as we continue to discuss unmet needs, is data aggregation to develop drug development tools. And the milestones to date have been the ongoing data aggregation, which takes time, and the continued expansion of our membership. We've drafted a consensus white paper to outline feedback from the LGMD task force members and come to a scientific consensus of how the data sharing efforts could accelerate the field given the data that we have in hand and where the white spaces are. We've introduced item response theory to the task force members in relationship to the NSAD, which Dr. Straub introduced as a multi-domain assessment tool, and how it may be responsive to different criteria, be it subtype, age, functional status, and whether that might be something that the task force members would like to explore in relation to how it could inform clinical trial design. The 2025 next steps will be to finalize that white paper, coordinate an in-person research roundtable with the members and the new onboarding members do an exploratory data analysis of to characterize those LGMD data sets that are in-house and do feasibility assessments of modeling plans for drug development tools. With that, I'd like to introduce our panelists who are prepared to answer any questions that may have come in from the audience. Please don't be afraid to ask questions. We have Doug Sproule from ML Bio. He is the Chief Medical Officer, and Dr. Jennifer Levy, the Scientific Director for Coalition to Cure Calpain 3. Kathryn Bryant Knudson, the CEO and founder of Speak Foundation. Cécile Ollivier, who is the Managing Director of the European branch of CPATH and also the VP of Global Affairs. Sophie Olivier, the Chief Medical Officer of Atamyo. And we appreciate you all for being with us. And at this time, I'm not certain that we have any questions from the audience, but please, audience, if you have questions, don't be afraid to ask. I would be curious to hear from Cécile, her thoughts on why this neutral environment that CPATH creates, whether it be for a task force like this one or a larger public-private partnership, is necessary to advance solutions to solve the various unmet needs of a rare disease space.
Yeah, thank you, Heidi, and thank you, Volker, for this excellent presentation. I'm very pleased to be here, and I'm really pleased also to see, particularly in the rare disease space, to have a joint EU-US venture to facilitate collaboration in accelerating the development in the LGMD space. And I think that one of the big issues with challenges around data first and then other development of tools such as endpoint or better disease characterization to minimize risk and uncertainties with evaluation of new medicines, for example, in this space, is the sustainability aspects you might have for some of the initiatives that are being conducted independently. And I think that that's just the way that some projects are, the funding mechanism behind it. They have a start and they have an end. And some of the challenges is that when some projects are ending, you need to restart and have some efforts, you know, to start again and to have a common space that is neutral, allow to maintain the sustainability and get the project going, like you can have several projects going, you know, in parallel. And when there is issues related to funding or other aspects that delay the project, you don't lose access to any of this data. And then when another project is being initiated by other partners in the same space, they can pick it up from where you left it, particularly because, and that's one of the advantages of CPATH, is that when you initiate this type of project, whatever that is at data level or new tools level, you establish regulatory precedents as much as possible, and therefore that allows to have this sustainability and to build up from one project to the other and truly advance the learnings and share the knowledge within the community and for the researchers. Thank you.
Thank you, Cécile. I think that's a wonderful way to answer that question, and how it the incentive for stakeholders to participate. I'm interested also in what current hurdles in the, we talked about how some of the LGMDs are ultra rare. What are some of the hurdles in very rare diseases as it pertains to international drug development? And how can working across rare diseases with small numbers and high clinical heterogeneity enhance data collection and innovation and efficiency, as you alluded to? for international populations. I saw COS already has set up, you know, their sites in a way so that they can do this. Sophie, if you'd like to take that one.
Yes, of course, with rare disease, there are multiple hurdles. But for LGMDs in particular, one of the biggest hurdle that I see, actually there are two. First, in clinical trial, you need, particularly in the early phase development, you need to enroll a homogeneous population. And with rare disease, because you have few patients available to enter into clinical trials, you need to restrict your population even further to make sure that you can see an effect after a reasonable time frame. So this is a big hurdle. I receive, and I probably Doug as well, receive a lot of emails from families aware of clinical trials available in the world, and they all want to enter into the clinical trial, but they do not meet the current inclusion criteria because as a sponsor, we try to enroll a very homogeneous population, the one that is the most likely to benefit for a curative treatment. And the second hurdle for me in LGMDs, in most of them, this is a slow progression of the disease. And of course, we have now a lot of potential endpoints that are sensitive enough, but sometimes we need to wait a long time before we can separate the natural evolution from the treated group. And this is where I think that artificial intelligence can be of huge help, because we can probably combine several endpoints and with different weighing of the endpoints to make some composite endpoints with artificial intelligence that would allow us to find a dose or a curative treatment and select, yes, a candidate much faster than if we have to wait 2 years, 3 years to see a significant difference in an NSAD or another endpoint compared to placebo or compared to natural evolution. This is for me the biggest hurdles.
Doug, I'd like you to have an opportunity to comment.
Yeah, I mean, to sort of build on that, kind of put it
Yes,
out in a different way, you know, from our perspective, obviously, I think one of the big challenges for this whole initiative is moving past a little bit of a, you know, mine proprietary view of you know, investigators who spend years building up a data set, working with patients, or industry partners such as ML Bio that invest a lot of money supporting this work, and having that ability to kind of let go and contribute in an appropriate way to this effort is really critical, and it drives development from a, you know, again, from a selfish perspective as a sponsor of a clinical trial, but also as a member of this community who's eager to see treatments emerge for all muscular dystrophies, particularly the limb-girdle muscular dystrophies, and specific to this conversation. The more data, the better, and that data can be specific. So, you know, we've contributed the data that we're able to contribute from the GRASP natural history study that we sponsored in LGMD2I/R9. You know, we obviously are hopeful that other industry partners and other academic partners will follow suit in the same sort of fashion. And this obviously has direct relevance for the development in LGMD2I/R9, for companies that are able to kind of amalgamate that data, but it has broad applicability across the space. You talked about ultra rare forms. All these are extremely rare. But when you're talking about some of the forms of limb-girdle muscular dystrophy that are listed in the dozens rather than the hundreds or the thousands of patients, the concept of doing a clinical trial is really challenging. So you have a drug that could work. How do you test it? And this is where this initiative and others similar to it can be really, really critical in being able to amalgamate data, understand where there's commonalities and where there's not, having as much data as possible so you can understand how to approach a trial, and potentially develop a valid and acceptable comparator arm so you're able to test a drug in as feasible a fashion, and get it into patients as effectively as possible. So there's a lot of potential benefits. There's the primary benefits, and then there's hopefully the secondary benefits that really bear fruit across the population, really accelerate the prospect of drug development. As a developer, if you have a therapy, that's great. You need to know how you're going to be able to show that it works. And this is where there's a critical hole, we all know, and this is one of this initiative has the potential to kind of help address some of that hole.
Thank you, Doug. That champions what we're trying to do, I think, with this task force wonderfully. And what I'd like to hear, both from Jen and Kat, a question came in from the audience. What do you think are the most needed tools for CPATH to try and develop for the LGMDs? Kat, maybe you can speak to what are the unmet needs that remain as a patient? And
Sure. Yeah, happy to. I think from the lived experience of the patients, myself, I'm a patient too, and then leading an organization and interacting with patients, the main thing that our patients tend to talk about is that they feel that they're replicating data all the time. They're in multiple natural history studies. We feel that we are recreating data sets, sacrificing blood, muscle tissue, time off of work, time off of school, the burden of trying to go to travel to another site. And we would like to see collaboration so that we don't have to continue to replicate data. I think companies also are in situations where they feel when they enter a space, and Dr. Sproule mentioned this just a minute ago, that they're having to start from scratch at times and create their own data set that they can utilize. And really those data sets for a good number of the LGMDs already exist for our community. And so I would just like to see that the patient burden is relieved by the efforts that we are participating in with the LGMD task force. I think if I could do anything here, it would be that in the future that our patients have a reduction in the travel that they need, and that also new companies can come into the space and more readily use a tool that's available to them and not have to create it from scratch. And I think that that would be very attractive in a pre-competitive environment to new companies. So those are kind of the things that I see are needs. I know Jen, she might have some answers there as well.
Just to repeat the question, Jen, it was, I wanted Kat to speak about the unmet needs from a patient perspective, but the audience asked, given what we've all been discussing in the task force and what's been presented, what do you think are the most needed tools that CPATH could address for the LGMDs at this stage? And we know they're data driven, right? So that we're working towards that.
Yeah, it's a great question, and Kat brought up some excellent points. I think from the field perspective, it's really about designing clinical trials that are effective and give unambiguous results. So, you know, in so much of what CPATH does in gathering this data is that we can understand what are going to be sensitive, reliable outcome measures. How do we set endpoints? What are the timelines that we're looking at? You know, I think, you know, the worst sort of clinical trial is the one where maybe it doesn't meet the endpoint, but then the patient community is saying we thought it worked, or there's secondary endpoints that don't agree with the primary endpoint. So I think, you know, being able to design those trials that get you the answer that you need is really critical. And I think the other thing I'll say that so many of the panelists have already touched on, but just about accelerating this whole process. There's a huge unmet need. People don't want to wait any longer than they have to. So anything that, you know, moves us faster towards that goal of getting therapies is super valuable.
And on the patient burden side, a question came in asking, and I know this work has been done and in the Duchenne space, can specific wearables be developed for the limb-girdle muscular dystrophies that can be worn by patients to gather data for clinical trials? So I'm interested both from Volker's perspective, because he works with the physiotherapists, and then also from Dr. Sproule and Dr. Olivier from the developer's perspective. What are your thoughts, Dr. Straub?
Yeah, so briefly, in Duchenne muscular dystrophy, we have now regulatory approval for an endpoint that is generated by wearables, the 35th centile of stride velocity. Now, this could also be applicable to patients with limb-girdle muscular dystrophy. The whole field is exploding, I have to say. And so there are so many companies that are now offering solutions about, you know, wearables and sensors that capture motor function, that I think we now need to apply some of this in clinical trials. We need to validate their usefulness. I would definitely see this coming for the limb-girdle muscular dystrophies. Again, the big question is, do we need to test it in each individual disease? Probably yes. Again, in terms of getting regulatory approval, because we want to make use of this in clinical trials and, you know, monitoring efficacy. So we are not quite there yet, but I definitely see what has happened in Duchenne will most likely also come to the limb-girdle field.
Yeah, just to build on that, I think what Dr. Straub hit on is one of the critical hurdles that, you know, as a community we can really come together to address, which is, as a developer, you want to be able to take the best measure that can show a meaningful benefit of a potential therapy for the population that you're studying. And this type of approach, where using wearables, has the potential to be really advantageous on a number of levels. It's not as dependent on how a patient is on a given day, but rather over an extended period of time. It is potentially a much more, you know, relevant and feasible and ease of use aspect, and obviously burden on patients a major aspect in any clinical trial. And it has a potential of being very sensitive to detect small but meaningful and important changes. The big challenge when I'm going to talk with a regulatory authority, I don't want the discussion to be about whether or not the measure is relevant, or whether it's applicable to this population, or whether it's been adequately studied, or whether it's, you know, it's a validated tool. I want the discussion to be over how this data that emerges from the use of this technology, the use of this endpoint, is impacted in the context of the clinical trial that we're conducting. And so that's one of the areas where, again, data amalgamation, working as a community to really push and advance these approaches with the regulatory authorities, such that as a sponsor, I'm not arguing about whether this is an appropriate tool. I'm arguing about how relevant the data that I'm generating is with regards to, you know, the regulatory needs of showing that a drug is actually effective. And so that's one of the holes, and it's a little bit of a chicken or the egg thing to some degree. But this is one of those areas where this can be an extremely helpful initiative as well.
And in a number of natural history studies for limb-girdle muscular dystrophies, wearables are now used already. So it's also important that it's not that we are missing this.
This group has such vibrant, wonderful conversations. We are now at time, so I'm going to let Dominique close us out. I apologize we couldn't have more conversation because I love talking with this particular group of stakeholders. There's always a number of topics.
Yeah. So thanks everyone for attending today's webinar. We'll be following up with a recording link for the YouTube video. And I hope everyone has a great rest of their day and a great holiday season.